Welcome to this beginning of the course on endocrinology. This is going to require that you read chapter 1 in the text, which is the Lang book, Endocrine Physiology. Now, as in all cases, I will probably cover certain things and put emphasis on certain things and not as much emphasis. But you are expected to have read the book, gone through the lectures each week, complete, whether it's case study or questions or whatever, by Friday of that week. I will grade them, and at the same time, we'll meet I'm looking at right now Saturday, for a Zoom meeting, which I invite all of you to attend. And I understand that some of you get very busy, even on weekends. And we will go over the case studies or the questions and take any time that you might have with just general questions, etc. So that said, let's go through some of the basic general principles that we are going to be dealing with in this course. Well, when we talk about endocrinology, we talk about homeostasis. And if you remember homeostasis, they say dynamic equilibrium. What does that mean? Well, it involves how do we balance and maintain internal function despite changes externally or internally. Now, when you think about it, the endocrine system does a lot of that. I mean, some of the key functions are the regulation of sodium and water balance and control of blood pressure and blood volume, exceedingly important, but also the regulation of calcium and phosphate to preserve extracellular fluid for cell membrane integrity and intracellular signaling. Aha, you might have thought, well, it's just for bone growth, calcium, phosphate, that makes for the bone. Yes, it does. But phosphate also it plays an important role as one of the buffers. Calcium plays an important role, not merely in just being the material, the element that you see so much in bones, but it's also, for example, an important ion, for example, with heart contraction. Okay? How do you get the synchronized contraction of a heart cell and then eventually having it synchronized with all of the other cells together? This is where we get into intracellular signaling. Also, we're going to see how endocrinology is playing a key role in the regulation of energy balance, control of fuel mobilization, utilization, and storage to ensure cell metabolic needs. We also have coordination of host hemodynamic and counter-regulatory responses to stress. Yes, there's a lot of factors that play a role, and we'll get into some of them. And finally, also, we'll see the regulation of reproduction, development, growth, and senescence. Now, you need to get used to some terms here. Probably you've seen these terms before, but it's important to go over them. When I talk about a hormone, I'm talking about a chemical messenger produced by an organ, released into the circulation to produce an effect on a distant target organ. A lot of times hormones are produced in very small quantities, and yet they have such incredible impact on the development of a variety of the target tissues or target organs. Now, endocrine glands, these are ductless, D-U-C-T-L-E-S-S, as opposed to the duct glands that you have with exocrine. I could make my joke now about, I don't want you to get confused ductless with duck quack, 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 and my Western Mass accent should be able to articulate that. But basically what you're doing with an endocrine gland is secreting chemical products. That's the hormones. But they go from the interstitial space and then they go into the circulation. There is no ducts, ports, if you want to say, tubes, etc. This is all basically secreted from the endocrine cell to the interstitial space, which is then picked up by the capillary bed around those cells, and they will carry this messenger of, or I should say this hormone really, throughout the entire tissues, organs, the entire organism. The term target organ refers to an organ that contains cells that express hormone-specific receptors that respond to hormones binding by a demonstrable biologic response. In other words, what I'm saying here is certain cells, if you know, are not going to respond to certain hormones. That's because they lack the receptors that will bind to that hormone, or they will not really be affected because the hormone can get through the cell membrane, but it's not going to bind to any intracellular receptors. We'll get into a little bit more about that when we talk about how certain hormones, such as thyroxine and the steroid hormones, how they're able to not necessarily need a receptor, but they will bind, but they get inside the cell because they are able to pass through the hydrophobic, that's the nonpolar region of the cell membrane. Now you have to keep in mind that hormone responses differ from neural responses. Why? Well, okay, let's take a look and do a comparison here. Message is usually an action potential. Endocrine, it's hormones. The nervous system will send this out via the central nervous system, originally the brain and the spinal cord. It will also have the cell types of the neurons, Schwann cells, glia cells. Those Schwann cells and the glia cells are important, especially to speed the progression of the action potential down the length of the axon. With endocrine, you have glands. They're specialized and produce these hormones. And they're a cell type called epithelial cells. Okay? Now, the transmission of the nervous system signals is usually by means of nerve cells called neurons. And usually you have entry into the target cell via the neurotransmitters at the synapses. Also, you're going to have openings of sodium and potassium channels. Now, with the endocrine system, the transmission of the hormone is going to be by blood vessels. Entry into the target, though, let's say the target cell, is either going to diffuse through the plasma membrane of that cell, or it's going to bind to receptors. Then we're going to get into eventually how that message then goes through, is transduced inside the cell, and begins to take effect biochemically in the cell. One of the big differences with a signal coming from the nervous system versus the endocrine system is the nervous system is always going to be short-lived. In other words, somebody basically dead stops in front of you as you're driving to class, boom! You know, you feel that movement and you make that quick decision, and boy, you stomp on that brake and you avoided an accident. But the endocrine system isn't always short-lived. Okay, nervous system we're talking about in some cases seconds, minutes. Endocrine system could be up to an hour before it really gets going, and it can be long-lived. In other words, secretion will extend itself beyond just a day, a month, a year. Okay? The nervous system, though, you want to keep in mind its control is unconscious or involuntary. In certain cases, it's conscious and voluntary. But the endocrine system is unconscious, involuntary. I don't have to think, oh, well, it's time for me to secrete T3 and T4. Oh, it's time for my islet cells and my pancreas to start releasing glucagon because I've been releasing insulin. You don't think about any of that. That just occurs. It is involuntary. It's part of the mechanisms physiologically that we maintain homeostasis. Now, sensory information. Well, nervous system does transmit and interpret sensory information. Not so with the endocrine system. Okay? So there's some big differences. If you want to sum it, here's the big key. Nervous system is going to be very quick on action potentials. You will have effects on various cells, but it will pass over a short period of time. Endocrine, hormones are being released. They pass through the general blood circulation. They will either bind to the plasma membrane, through the plasma membrane into the cell, and bind to certain selective receptors, receptor proteins inside the cell to initiate certain biochemical changes. Could be genetically affected or just basic biochemistry in the cytoplasm of the cell. Let's move on. When I talk about hormone chemistry, okay, this is where we get into this. If you've ever had biology or biochemistry, sometimes it's almost like, well, are we talking about is it a protein or a peptide? What's the difference? We all know that the basic building block of proteins and peptides are amino acids. And if you read your books, sometimes you'll find that they use these terms, protein and peptide. Well, isn't a peptide a protein? Yeah, but what happens is usually people will designate. A lot of researchers will just designate it being a protein if it's about 50 amino acids or longer. If it's under that, they define it as a peptide. Still basically in the protein grouping, it still has amino acids as its building blocks. Now, a lot of the hormones, the majority of them, are protein or peptide based. They range from 3 to up to 200 amino acid residues. Usually they're synthesized as what we call a pre-pro hormone. In other words, think of it as a much, much longer piece. If you want to think of just beads on or pearls on a necklace or beads, this is where we get into what we call the post-translational processing. Certain enzymes will be activated. They will clip and cut off certain pieces. And some of those pieces will be the peptide hormone of focus. Others will be sort of floating around. It's interesting because some of them are incredible. We're going to get into POMC. It's called pro-opiomelanocortical hormone. It's this big, long chain of amino acids. And literally, depending on where you put the cuts, you'll get endorphin and enkephalin, melanocyte stimulating factor, ACTH, and a bunch of other ones. Don't worry as much about that right now. We'll get into those when we get into certain sections of the text later on. But what is important also is that these peptide hormones, they're secreted into these structures called secretory granules. They're membrane bound. And before they're being released by exocytosis, the release is going to be similar to the release of neurotransmitters from the nerve terminals as exocytosis. You're going to have some sort of chemical signal, some sort of physical signal, something that's going to occur that will cause the release of these granules by exocytosis, and they're going to dump their contents outside the cell. These contents are the hormone, and they will eventually be picked up by the blood capillaries. And you've got a variety of these protein peptide hormones: insulin, ADH, antidiuretic hormone, oxytocin, glucagon, ACTH, TSH, LH, FSH, HCG, human chorionic gonadotropin. We'll get into that later. And you also have some of the releasing factors, which we'll get into next week, which come from the hypothalamus, and some of them are really, really short, about 3 amino acids long. Now, you also have to keep in mind something else. Proteins are sometimes, I would call it, decorated. What I mean by that is they are synthesized and then go through processing at the rough endoplasmic reticulum, and in some cases also at the Golgi complex, or the Golgi bodies. And they have carbohydrate moieties. This makes them being designated now glycoproteins, in other words, sugar molecules that are added on in certain places. This is to help the hormone achieve proper receptor binding and also circulating clearance of the hormones. Now, amino acid derived hormones, how do they differ? Well, really what you're talking about is taking one single amino acid, and then you biochemically modified. Sometimes you're going to get what they call dimers. So in other words, two of the very same amino acids, in the case we're going to use as an example, is tyrosine. They're bound together. And this is basically the building block to make thyroid hormones, T3, T4. This is important because they're insoluble in blood plasma, yet they cross the cell membrane. Now, you might sit there and say, but wait a minute, Dr. Roberge, wait a minute. They're insoluble in blood plasma, so how do they get transported? This is where later on we're going to talk about this. There's going to be certain blood proteins, albumin, and a whole host of other ones that act as transporters. They kind of allow these hormones to piggyback and move along, okay, throughout the bloodstream, and then they're released, and they basically are able to easily cross the cell membrane. Now, what are some of these amino acid-derived hormones? They include catecholamines. Catecholamines are usually modified phenylalanine or tyrosine. For example, how can PKU patients, which are phenylketonuria, tyrosine? So they just have to make sure that they consume tyrosine, which is easy. And then these are biochemically altered slightly to dopamine, epinephrine, norepinephrine. Now, all catecholamines are soluble in blood plasma. These type of hormones will attach to cell receptors. A lot of them are modified by various endocrine cell enzymes, okay? And this is where we get this modification. To help you, what you have is tyrosine. It's converted over to L-DOPA. You probably have heard of that as a drug that's used to treat patients like with Parkinsonian symptoms. And L-DOPA then gets converted by another enzyme to dopamine. Okay? We'll continue on this later on in detail. Steroid hormones. These are derived from cholesterol. You're going to see them in plasma or the blood, period. The advantage here is just because they're lipid soluble, they're cytosolic or nuclear receptors. Now, when I talk about this, you might just think of testosterone and estrogen. No, there's more than that. There's aldosterone, cortisol. There is the vitamin D, which is also known as calciferol, as well as testosterone, progesterone, and estrogen. We'll get more into those in later subsequent chapters. But I wanted to give you a kind of a view of what you could see for some of these examples. Here is norepinephrine. This is an amine hormone. Here is the peptide hormone oxytocin. If you notice here, what you've got is really about 9 amino acids. That's a short chain. And then we have steroid hormones. They're all derived from lipid cholesterol. If you'll notice, the difference between testosterone and progesterone is not really that significant. I mean, here's your ketone structure here, and here is a methyl group. Here's another methyl group. The big difference here between testosterone and progesterone is this carboxylic acid group here, or this hydroxyl group here. Now, you don't have to lose sleep of whether I'm going to tell you, is it on this or that, etc. When I tell you that certain things you will need to know for exams, yes. But I would like you just to be familiarized at this point with some of the basics, and that is that if you have a steroid hormone, the basic structure is that 4-ring structure here that you see, which is sort of the cholesterol is the foundation. And what really differentiates various types of steroid hormones are these functional groups that you see attached on the sides here. Okay? Now, there's a number of molecules that act near the point of hormone synthesis and are included in the class of paracrine hormones. We'll get more into what paracrine is in a minute. Eicosanoids also go by the nickname local hormones because really what they do is they act on cells very close to the site of their own production. Eicosanoids are also rapidly, they break down. That's why, in a way, it makes sense. They're only going to be able to go so far from their site of production, but then they are able to eventually just break down. So they really wouldn't be able to travel far, let's say from release, let's say in your brain, all the way down to, let's say, your toes, because they've probably broken down in transport. There are three most researched types of eicosanoids. They are the prostaglandins, the thromboxanes, and the leukotrienes. Now, eicosanoids are derived basically from arachidonic acid and related polyunsaturated fatty acids. These are called PUFAs, P-U-F-A-S. I know it sounds kind of funny, but one of them is best known as eicosapentaenoic acid, EPA. Some of these also show up in importance because of the fact that when we talk about uh, early childbirth and early child brain development, first year or so, the milk that has been given, or whatever you want to basically say, in other words, the formula, that's what I was trying to find the word, given to babies, they found that if they give them certain types of arachidonic acid or polyunsaturated fatty acids of particular types, this enhances brain development and brain growth. Now, if you take a look here on these charts, I basically have taken, here's an example of prostaglandin E1. If you notice, they all start off with arachidonic acid. Notice where my pointer is here, and it's a big long chain with sort of a bend here. Remember that these are double bonds here and here and here. And what happens is that it's enzymes that cause the changeover and the formation of unique structures. In this case, you have prostaglandin E1. It is also goes by the designation PGE1. All prostaglandins will start off with a capital P, capital G, and then usually you get into the last designation, which designates their unique molecular structure. F1 alpha, E1, as you can see, are just two examples, all right? Notice that they all have, many of them have this unusual formation of this pentagon structure right here. Now, if you'll notice also the difference between the prostaglandin and the thromboxane, and notice it's A2, which obviously means there's got to be an A1, and a few others probably. Like I said, they've been most investigated, but they're still finding out even more stuff about these particular unique types of hormones. Oxygens have been moved into the rings, as you can see here. So there's been some replacement of the double bonds with oxygen attachments. And then we have the other group called leukotrienes, and this is B4. You'll notice that there are three conjugated double bonds, here, here, here. You'll also notice that there's one over here too, which I suspect was previously arranged from the original arachidonic acid. The key point here is this. We start off with arachidonic acid, okay? And then we have these various enzymes here that will convert it into either thromboxanes, leukotrienes, or prostaglandins. Now, you might sit there and say, boring. It's not. When you think about it, thromboxanes play a key role in allergic reactions, particularly with asthma, etc. Prostaglandins play a key role in wound injury and inflammation. Bad bruise, you sprained your muscles, you were doing some exercise or something. What do you usually do? Well, you go run off to the aspirin or to the other types of non-steroidal anti-inflammatory drugs like Motrin, which is really the type of drug that also is acting against the formation of these prostaglandins. And then we have also naproxen. And now we have Mobic. Now, what I'm trying to say to you is that basically what we are doing is using drugs that interfere not with the actual prostaglandin itself or thromboxane, but it blocks the enzymes that would cause this conversion. So you don't have the continuous production of that prostaglandin at that damaged muscle site or at that sore arm site. Okay? That's what those drugs actually do. Now, we're going to get into the type of hormone effects. We have endocrine, paracrine, autocrine, and intracrine. The best known are the endocrines. That's the hormone that is released into the circulation. It then travels in the blood to produce a biological effect on target cells distant from the site of the hormone release. An excellent example. Antidiuretic hormone produced in the posterior pituitary. Where does it act on? Well, it can act, it's also known, by the way, by the name vasopressin, but it does play a key role in bringing back water from the filtered urine that is being, or the filtrate that they would call it, that's being produced in the kidneys. Now, that's a distance away from the original site of its release to the target cells. Paracrine, this is a hormone released from one cell that produces a biological effect on a neighboring cell, frequently within the same organ or tissue. An example of this is that when you look at an islet of Langerhans, these are the cluster of cells that produce glucagon, insulin, somatostatin, and pancreatic polypeptide. These cells not only release the glucagon and insulin, but we also see evidence that the somatostatin seems to play a role in balancing out the release of insulin and glucagon. And that would be a paracrine example. Autocrine, well, just like feedback, that's an easy one, is a hormone that produces a biological effect on the same cell that released it. So it's almost like it's a self-regulatory mechanism. Hormone is produced from a set cell, but as it's being released, that hormone has an effect to either positively or negatively affect the continuous release from that same cell. Now, intracrine, the hormone acts inside, intracellularly, in the same cell. We're going to mostly focus in on the endocrine action, obviously. But autocrine, here you can see this, a release of that particular hormone, and yet it feeds back on the autocrine. Paracrine, to a nearby cell. Remember that we talked about the eicosanoids. They can pretty much be considered having a paracrine action by nearby cells only. The endocrine, this is clearly where you get into effects that travel quite a distance from their release to the target cell. And what's so neat about this is that it's just using the blood circulation. Now, here's another chart which I thought would be interesting. When you see the paracrine effect, here it is. You have a particular protein here, and it's going to act on the receptor of nearby cells. And this is a gene product, but I wanted to grab this particular image because I thought it would be helpful in clarifying this. So you have this hormone, or you have this protein that comes out, but as soon as it comes out, it has an effect that modulates the activity of this cell in releasing the very same hormonal product. Then endocrine is something where it's believed that the amount that's produced may find its influence on receptors inside of the cell itself, or actually back on the genetic expression on the DNA of that particular hormone. Now, the next thing we're going to talk about is hormone transport. Hormone transport depends on water solubility. Yeah, we know that. We've been talking about blood plasma for a while. That's true. That's true. Many hormones circulate very freely. Now, some hormones circulate, and basically they're bound to carrier proteins. Now, this is important because it serves as a reservoir for the hormones, but it also extends the hormone half-life. One of the things you have to understand is that when we have hormones in the blood. The blood is not just red blood cell, white blood cell, thrombocyte, water, some ions. It's got a lot of other important materials in them, substances in them, antibodies, protein carriers, enzymes in some cases. And therefore, they're going to affect the time period that the hormone is going to be lasting in the blood. That's where we're going to get into this concept of half-life. And that's really the time the hormone decreases by 50% from its original concentration. Keep in mind carrier proteins. Most carrier proteins are made in the liver. These include the globulins, they include albumin, they include specific binding proteins for cortisol, thyroid, and androgen hormones. Here's one thing to keep in the back of your mind. Especially if you're thinking about medicine down the road, you might say to yourself, okay, we know that the liver is involved with filtration and detoxification and all that. But what might be one other indicator that the liver is beginning to fail? Perhaps its production of these key globulins, which would then cascade into problems clinically in measuring the types of hormones out there. It's not that the person is not making the hormones, it's they're not showing up in their carriers because the liver is not producing as much of the carrier proteins necessary. Just something to give you to think about. Remember we talked about dynamic equilibrium? This is the interaction between a given hormone and its carrier protein. Now, in this case, what we're talking about is the capability for adjustments to prevent clinical appearances of either deficiency or excess due to changes in binding protein levels. So in other words, the amount of hormone that's out there can be carried by these carrier proteins. And if you produce way too much, you may not be able to have it really being present in the blood because you've saturated all of your carrier proteins. Consequently, if you don't have enough, you might see a lot of carrier proteins that don't have any hormone bound to them. Something to again think about. Now, when we talk about metabolic clearance rate, this is the volume of plasma cleared of the hormone per unit time. Now, what do you mean by that? Well, basically what happens is this. In metabolism, as I've said before, in blood, you're going to find that there's a variety of enzymes and antibodies and other things. And once you start breaking down some of those hormones, the levels will drop. Now, where are you going to see specific effects? Well, that could occur in a variety of organs. After the hormone binds to a specific receptor of the target organ, you have to keep in mind that the hormone clearance will occur via two routes. The metabolic transformation, that is the inactivation by the liver. And there's two phases, and yes, you do need to know these. One of them is hydroxylation or oxidation. So basically, there is enzymatic effects that inactivate the effectiveness of the hormone via hydroxylation or oxidation. Phase two is glucuronidation, sulfation, or reduction by glutathione. All right? The hormone will be excreted by the liver through bile or via the kidney by urinary excretion. Similar to what you talk about when you think about pharmacology. How is the drug inactivated and then excreted? Is it purely by bile, which would be out the feces, or is it done by urinary excretion, or is it part this, part that? You also have to keep in mind that some hormones are degraded via hormone receptor internalization, followed by lysosomal degradation. You see that very commonly with insulin. But you want to keep in mind there are other routes. And it only makes sense when you think about it because hormones are really effective and really present in extremely small amounts. So why is it that I don't have steroid molecules testosterone, 20 years later rolling around inside me because of these principles, basically of the neutralization, the breakdown of them. When I talk about the biological response to hormones, this requires the binding of the hormone to the receptors at a target organ. Okay, fine. Hormones circulate in very low concentrations, 10 to the minus 7th to 10 to the minus 12th molar. Therefore, that receptor must have a high affinity and specificity for the hormone to produce a biological response. All right? Keep in mind, when I talk about specificity, that's the ability of the hormone receptor to discriminate among other hormones with related structures. Okay? When I talk about affinity, it's determined by the rates of dissociation and association of the hormone complex under equilibrium conditions. You might sit there and say, I think I understand what you mean. Well, think about it this way. We'll make it a real simple example. We have hormone X. Now, when hormone X gets sulfonated, oxidized, etc., how is it that it doesn't bind to the receptor at the target? And what you want is a very high affinity and high specificity so that no other molecules could be confused and give a false signal to that tissue. We have what we call an equilibrium dissociation constant, HD. This is defined as the hormone concentration required for binding of 50% of the receptor sites. Now, here's the key point. The lower the KD, it shouldn't be KD, it should be HD, I'm sorry. The lower the HD, the higher the binding affinity. In other words, affinity reflects how tight the hormone receptor binding is. And what you really want, keep this in mind, is you want an HD that is really, really tight. And therefore, it's not going to have it attach and detach, attach and detach. If any of you remember your biology, one of the things is that you have to get away from just the turning the switch. I come into a room, it's dark, I flip the switch, it's on. With certain molecules to the receptors, they will bind, detach, bind, detach, bind, detach. Every time they bind, you get a signal that's sent into the cell. This is with the receptor. But that's more of what we have in a biological situation, not flipping a switch, but association, dissociation, association, dissociation. I hope that makes it clear. Biological response to hormones requires the binding of the hormone to the receptors at the target organ. Yeah, we said that. Now, you want to keep in mind something. Some drugs can simulate hormone binding to the receptor with very positive biological effects. These are called agonists. Okay, I get that. So what about the drugs that stimulate hormone binding to the receptor but have negative biological effects? They block the receptor. These are called antagonists. There are some drugs that we use to replace the actual missing hormone. And these agonists are worked out in some ways. And I'm not talking about insulin now for diabetics. Because with insulin, that's the actual hormone. But some other attempts, for example, I'm thinking of leptin as an example, they're all trying to find ways to make a drug that would bind to the leptin receptors, having a positive effect, which would mean that you would reduce eating. Anyways, we'll get into that more later on. Here's one of the big problems that exists, and you can see this in my slide. Mutations can alter the binding specificity or affinity or the hormone receptor for the hormone, and hence alter the biological response or inhibit it. What did he just say? I know it sounds a little confusing, but let me put it to you straightforward. Now, in the 1990s, scientists were able to understand and isolate a hormone that was made by the adipose tissue. It's called leptin. And what they did was they used a population of exceedingly obese individuals. And they found that one group had extreme obesity. Leptin would be the signal, it's the hormone. And when the leptin levels rose in normal people, they would stop eating. In some of these extremely obese people, they weren't making any leptin. Now, there was a second group that had all the physicians scratching their noggins for a while, going, wait a minute. I got this other group here of extremely obese people, but they've got super high levels of leptin. And that's when they were able to isolate and discover the leptin receptor. It turns out that with high levels of leptin, it's usually the defect is not in making the leptin. The defect that leads to the obesity is in the leptin receptor. Think of it in a simplified way. You have a radio, you're broadcasting. Now, if you were not broadcasting, no one would get the signal. But if you were broadcasting, but no one had the receiver to hear your signal, then you would still have the same communication problem. The difference is whether it's the hormone or the hormone receptor. Individuals in some cases that have defects not in making leptin, but in the leptin receptor. And the leptin receptor either somewhat or completely does not bind to the leptin. I hope that helps. Now, we have to start thinking about hormone receptors are classified depending on their cellular localization. So we talk about the cell membrane, and we have these very interesting structures called intracellular receptors. They're basically proteins that span the entire width of the cell of the lipid bilayer. And they will have the hormone bind to that receptor on the outside. But then we've got to get that somehow that signal inside, because the hormone can't get inside that cell to cause biological changes. So this is where we're going to get into some of these details. And this is to keep this in mind. This is what separates out thyroid, steroid, and peptide and catecholamine hormones. See, peptides and catecholamines are unable to cross the cell membrane because of that lipid bilayer. Okay, what's in the center of the lipid bilayer? It's all hydrophobic, the fatty acid tails from the phospholipids. But the peptides and catecholamines bind to the cell membrane receptors. Now, with thyroid hormones, they are transported into the cell and bind to a class of receptors called nuclear receptors. They kind of form this complex where the thyroid hormone binds to the receptor, and then they are able to go inside and go inside of the nucleus and affect gene expression, gene transcription, turn genes on, turn genes off, etc. Steroid hormones are lipid soluble. They are easily able to cross the plasma membrane, and they bind to intracellular receptors. They also may bind to membrane-associated receptors, too. Now, when we talk about a hormone receptor complex, this is going to initiate a signaling cascade of the intracellular events resulting in a biological response. Because when you think about it, well, look, catecholamine, or let's say ACTH, it stays on the outside of the cell. So how do you get the changes going on inside of the cell? That's where you have what's called the secondary messenger system. Now, these are divided into ligand-gated ion channels, or receptors that regulate activity of the intracellular proteins. Let me take the smaller one first, ligand-gated ion channels. These are basically similar to what you've seen with acetylcholine at the neuromuscular site. You have the neurotransmitter bind, and the ion channel opens up and causes a change in the polarity of the postsynaptic membrane, or the cell. That's the target organ. A lot of times, hormone binding to the receptor will produce a conformational change. In other words, remember that proteins can flex, and many of them flex in such a way that they will open up a certain ion channel on the cell membrane, and that'll cause, within seconds of the hormone binding, you'll have this flush of ions going either out or in, depending. This depolarization may trigger other biochemical effects inside the cell. Now, receptors that regulate activity of intracellular proteins. This is going to be a little bit more important, and you need to spend a little time on. Best known are the G protein coupled receptors. We also have receptor protein tyrosine kinases. And then we have also the class 1 cytokine receptor. This also activates intracellular tyrosine under the Janus kinase JAX2 system. And so this activity will phosphorylate tyrosine residues, and some proteins directly, they're going to be inducing physiological effects. And basically, you see this with a lot of cases of growth hormone and prolactin. Now, in this secondary messenger system, it really depends on the receptor, the ligand that binds to it. It really depends on what type of G protein as well. Now, we'll work this out here a bit. Hormones that use basically these secondary messenger systems for this method of signal transduction include thyroid stimulating hormone, glucagon, vasopressin, ACTH, LH, FSH, and catecholamines. Now, G-coupled receptors, usually what you have is a single polypeptide chain with 7 transmembrane domains. In other words, basically it's going back and forth across the membrane. And then that's coupled with a heterotrimeric guanine proteins, G proteins. And they consist of 3 subunits, alpha, beta, and gamma. Now, hormone binding to the receptor will trigger the interaction with the G proteins. This results in an exchange of GDP, for it also will activate the G protein. The activated subunit is released from the receptor, and then the alpha subtype is released from the beta gamma complex. There are all these little complexes together. Now, when we talk about cell membrane receptors, there's 2 main enzymes that interact with these G proteins, and the exact interaction is controlled by the G protein. This is where we get into what we call the cascade effect. All right? And so this is going to be either adenylate cyclase, and you're going to see this in the diagram. You also will see it in your book. The responses can result in an increase or decrease in the making of a unique molecule, cyclic AMP. Start with ADP, and you basically make it into a cyclic AMP. The other type is phospholipase C, which is an activation of that. Remember I said that there were several different subtypes of G proteins? Now you might sit there and say, why is this so important? Well, it turns out that some of them play some absolute key roles, and we can direct drugs to act on these G proteins. Okay? Now, there are 4 families with different effector proteins. There is G alpha S, and rather than have that little s squiggle down, we had G alpha. Okay, I'm back here. I'm sorry about the break there. I had to make some corrections here. By the way, the earlier one, the HD with that formulation for binding is really KD. So I've made that clear. Let's get back to G proteins. There's subtypes. There's 4 families. G alpha S activates adenylate cyclase. So you're going to see this increase in adenylate cyclase, which means that you're going to have an increase in cyclic AMP. Now, G alpha I inhibits adenylate cyclase, which means that you're going to have a decrease in cyclic AMP. G alpha 12 activates Rho guanine nucleotide exchange factors, which basically go right into and act on genetic expression. G alpha Q activates phospholipase C, which we'll get into in a second. I do want to bring up one other thing. Cyclic AMP is broken down by phosphodiesterase. Okay. So you probably had a coffee if you're a coffee drinker. And you're probably sitting there going, yeah, I'm getting into this sort of. What stops phosphodiesterase? A variety of methylated xalenes, including caffeine. That's why caffeine gives, in part, the boost that we want, because basically what it does is it inhibits that enzyme, and so the signal for cyclic AMP lasts longer physiologically. Okay, now, by the way, if you want to review this, go to page 9 in figure 1.5. You'll see this well done out. Let's go to phospholipase C. What is that? Well, this is another system that occurs inside, this cascade for a signal. And this is basically results in the hydrolysis of phosphatidylinositol bisphosphate and the production of diacetylglycerol, DAG, and inositol triphosphate, IP3. Now, DAG activates protein kinase C, which phosphorylates effector proteins. IP3 binds to calcium channels in the endoplasmic reticulum, leading to the release of calcium ions into the cytoplasm. Sounds a little bit like a muscle, isn't it? Well, remember that the endoplasmic reticulum, its comparable structure that's similar is the sarcoplasmic reticulum in the skeletal muscle. The phosphorylation of effector proteins and cellular responses. Now, you can see this. Remember we talked about that 7 transmembrane structures here, and you can see all of these different types of hormones and other stimuli, light, odor, and pheromones, etc. This is very common, what you're going to see. But obviously, all of this, the biogenic amines, the amino acids, ions, lipids, etc., they can't get inside the cell to really fire up and say, go, do it. So you have this cluster here of alpha, beta, and gamma. Now, once we have the activation here, we're going to free up the basically the alpha GDP molecules. Excuse me, I made a correction again. It's a GTP molecule, not having a GDP to GTP. Not having GTP to GDP. It's GTP activated to GTP, meaning guanine diphosphonucleotide converted over to guanine triphosphate nucleotide. I want to make sure that's clear. Like all professors, we make a mistake. You want to correct it and make sure everybody's clear on the thing. Notice this cluster right here. This is the beta gamma. Now, these would be activating adenyl cyclases, ion channels, etc. But it's these 4 families right here that I mentioned to you earlier. Okay? GI, adenyl cyclase, turned down. Inhibition of cyclic AMP production. GDP alpha Q, that's basically where you're having the phospholipase activation. Alpha S, you have adenylate cyclases turned up and therefore you're going to have an increase in cyclic AMP concentration. And finally, we have alpha 12, which really works on Rho GEFs and Rho, and these are going to play a role on genetic expression. That's all you really need to know at this point. So we got to a point and we had to bring up one other area, and that was receptor protein tyrosine kinases. Keep in mind that all of this that is being discussed is basically how is it that a signal that, like a hormone, that is stuck on the outside of the cell, how can we change the biochemistry of what's going on inside the cell? Okay, here's another route. Usually, receptor protein tyrosine kinases are going to be a single transmembrane protein, and they're going to have their own intrinsic enzymatic activity. These hormone receptors, including the system, are going to be examples of insulin and insulin-like growth factor. Okay? The hormone binding to the receptor activates an intracellular kinase activity. So you have the binder on the outside binding to the hormone, and it passes, basically that whole structure is passing into the inside of the cell. And it is going to activate that enzymatic part, the kinase activity. It's going to result in phosphorylation of tyrosine residues in the catalytic domain of the receptor, which leads to the downstream cascade of proteins recruited and activated. So it's going to be basically binding of the insulin, activation of this particular protein here, and you're going to start doing this where it becomes phosphorylated, and when it's phosphorylated, it's going to start acting on other basic proteins that are going to be activated by their own phosphorylation. We can see that here. Okay. Now we get to the other part. Remember all those ones that we couldn't figure out before, the steroid hormones and vitamin D and etc. How do they get inside, and what do they do? This is where we have intracellular receptors. Receptor family that binds to the hormone, then as a complex binds to selective sites on the DNA to act as a transcription factor. They either activate or repress the gene expression. Keep that in mind. It's not just always turning things on. You have to also be able to turn genes off. So what are the requirements for this intracellular receptor? Well, the hormone that you're going to be dealing with must be hydrophobic. It either crosses the plasma membrane or it is actively transported into the cell. So what hormones are we looking at? Steroid hormones, vitamin D. Now, you also have to deal with unbound receptor locations. They're either going to be solely in the nucleus, or they're going to be out in the cytosol, and they may be eventually then transported as a complex of the hormone and the cytosolic receptor and brought into the nucleus. Now, the hormone receptor regulation, the hormones influence the responsiveness to the target cell. Target cell must modulate the receptor function and density of receptors over a wide range of stimulus intensities. Therefore, the cell must have the ability to undergo a reversible process of adaptation. What you have to think about is this. If you keep this in mind, target cells will have a variety of receptors there. Even if we are not producing the maximum or the minimum required amount of hormone production, what amount is present will meet up with some of the receptors, will bind, will get transported and activate on the cells. But at the same time also, the cell will have the ability to undergo a reversible process of adaptation, which means what? It means that you're not making excessive amounts of receptors for no reason at all. I have taught this many times in my biology classes. Nature hates waste. Okay? So you're not going to make lots and lots of receptors if you only need a small amount to. This gets us into also a couple of other avenues: desensitization and upregulation. When we talk about desensitization, this is also known as receptor downregulation. This is a process where prolonged exposure to the hormone decreases the cell's response to that hormone. This happens a lot. Cells that get whopping amounts of it will slowly, progressively reduce the number of receptors they have in that cell to basically self-regulate themselves in some cases. Now, of course, this includes the process of inducing endocytosis and temporary sequestering in endosomes. Endocytosis with receptor destruction by lysosomal enzymes can occur. You can also have desensitization by the receptor protein phosphorylation. What about the opposite? A reversal process called upregulation. That's to increase the sensitivity to the hormone by increasing the number of receptors on the cell surface. So we have these means of either decreasing or increasing the number of receptors. We don't overproduce receptors. We don't underproduce receptors in nature. And what I mean by in nature is there's no gene defect, there's no cells going cancerous, there's none of this. But if you notice here, what you've got is a ligand binding to a receptor. It's brought inside the cell by this clathrin-coated pit. This is basically an endocytosis. So the clathrin will depart from the vesicle. The receptor and the ligands will eventually separate. And so you have this endosome that kind of pinches out the receptors. Now, there's two things that can happen. Obviously, the receptor can be destroyed, or they can be recycled. And that's where you see this happening, where they will eventually have this vesicle bind to the cell membrane again and then be, how can I say it, displayed out here now to be reused. Now, in downregulation, you'd probably reduce the amount of receptors. Upregulation, you might increase the amount of receptors. Okay? What happens to the ligands? They go to the lysosomes or the Golgi for processing. Okay, remember lysosomes contain those acidic hydrolytic enzymes that will break down the proteins, etc. Now, another thing, secretion of hormones requires synthesis or production of the hormone and the release of the cell. So, does that mean it's like turning on a switch? Nope. You have to keep in mind that plasma levels of hormones may oscillate throughout the day, the month, from day or night. Their release may be influenced by photoregulatory factors such as light. Factors that influence the release include hormone releasing factors, okay? Because you have hormone releasing factors, then you have the hormonal stimulating factors, and then you have eventually the target organs such as the thyroid or the adrenals, etc. And they will release their hormones, which will then go to their target organs. Now, another way to look at it is neural, and this is a direct neural control via the hypothalamus. The hypothalamus, you have, for example, this connection between corticotropin releasing hormone and adrenal corticotropin hormone. The adrenal gland in the adrenal medulla, you have the epinephrine that's going to be released. Remember, that's part of the fight or flight system, the sympathetic autonomic nervous system versus the parasympathetic autonomic nervous system. Okay? Now, you're going to have some means that will affect releases of other hormones because of epinephrine release. All right? What are some of the other factors that influence release? Besides neural, besides hormonal releasing factors, we have nutrition. Huh? What? You kidding? Yeah, because plasma levels of glucose going up, going down, they influence or trigger the release of certain hormones such as insulin and glucagon. Water. Too much, too little, and that can be played on by a role by antidiuretic hormone. Sodium. and potassium ions. These are all going to be influenced through the aldosterone, natriuretic factor, which also is known as natriuretic peptide. And all of these, the levels which shift, water, sodium, potassium, are partly regulated that are constitutive, that is basal levels versus stimulated levels. Now, what I mean by basal is you don't do anything, you're just resting at night, and there's a certain level that's always there. Then suddenly you have a situation that's a high stressor, so you're going to have cortisol being massively released. So that would be the stimulated peak level example. Temperature, fight or flight, rest and digest. Fight or flight, rest and digest is the sympathetic, parasympathetic. If you take a look in your book, you're going to see a couple of really cool charts. One of them is on page 16, and what that does is it gives you patterns of hormone release. Sometimes what is going to be the timer for all of this is going to be something as simple as light. By the way, if you notice here, this is an example of what I was saying before earlier about this diagram using upregulation of the receptors. Okay, so we've got a binding here, and what happens is just one binding is going to trigger more receptors being made, and even more receptors made because there's not enough hormone receptor binding that's occurring. So the cell will regulate itself and increase the number of receptors to grab further hormone molecules. Down regulation, we have an excess here, and you can see there's a lot of the receptor and the hormone binding, and so the cell begins to remove the number of receptors on the outside. You still have some higher amounts of binding that goes on, and eventually what you get is a reduction in the number of receptors until you have a basic level that the cell can accept without overstimulation or other unusual activity. As I mentioned to you before, the fight or flight episode, well, let me bring up something. Let's go over this here. And this might have been a repeat slide, so bear with me. If you look at hormonal control, we've talked about light, we've talked about nutrition, we've talked about neural impulses, but we also have to have these feedback mechanisms. Now, if you're familiar with feedback mechanisms from anatomy, physiology, remember that to obtain homeostasis, you will have negative feedback systems and you will have positive feedback systems. Now, one of the best systems or best examples that I can think of right off the bat is this relationship called the HPA, the hypothalamic pituitary axis. Because really what you have is endocrine organ. In this case, for example, right here, we have the thyroid. The thyroid will, of course, release thyroxine, which will affect target organs throughout the body. Okay? But how do we regulate this so we don't keep producing too much or not enough? That's where we get into negative feedback. The purpose of negative feedback, really, with negative feedback, you're reducing the response to this initial stimuli, where you're reducing the stimuli itself. Now, negative feedback by thyroxine will affect the secretion of TSH, thyroid stimulating hormone, as well as thyroid. They made it into this very long one, but it's also known as thyroid releasing hormone, or TSH, thyroid stimulating hormones releasing hormone. When elevated levels of thyroxine occur here and here, these hormones, the TSHRH and the TSH, will decline. And as they decline, that will reduce the amount of thyroxine released into the body. That's negative. That's a negative feedback that I've shown you. Now what we're going to do is show you basically nutritional control. Nutritional control, you can see really simply. You could have this as glucose with insulin and glucagon. When I have elevating levels of glucose, I have insulin that increases so that it can drive the glucose out of the blood back into the cells. When I have plummeting levels of glucose, I have the release of glucagon, which then mobilizes glucose to elevate its levels in the blood, and that's usually by breakdown of glycogen in the liver and also some mobilization of other resources to act as cellular fuel. All right? But we're just focusing just on glucose right now. The same thing occurs with calcium ions. You have PTH, which may mobilize the amount of calcium in the blood. Whereas if you have too high of a level, you have the opposite. Calcitonin begins to take into play, and you have the sequestering of calcium, for example, into the bone structure. Now, there's more than that is going to be occurring. But when we get to that area of calcium regulation with PTH and calcitonin, we'll get into more details. I just want to get you into a basic concept of this. This is all, but also in the male, that there's going to be more than just the amount or response of testosterone that will have a negative feedback. There'll be inhibin and a variety of others. Let's use a terminology here, stimulus. This is the factor that initiates hormone release. Then we have negative feedback regulation. We've already been talking about that. Hormonal inhibition of hormone release, and it's triggered to reduce the stimulus. But what about positive feedback? This is hormonal stimulation or enhancement of the original stimulus. Where do we see that? Well, we're going to get into that in a second. There's only a few examples that are very clear with positive feedback, but I thought that this was a nice chart to give you this comparison between negative feedback homeostasis and positive feedback homeostasis. In negative, the feedback mechanism results in the inhibition or the slowing down of a process. In positive, the mechanism is resulting in the amplification or growth of the output signal. Negative reduces the effect of the stimulus, decreasing productivity. Positive bolsters the stimulus, increasing productivity. Positive, there's a lot less frequent that you're going to see for examples. Two of the best examples that everyone describes for positive feedback are either the blood clotting mechanism or childbirth in mammals. Negative feedback is much more prolific, and you're going to see this in control of optimal internal environments for blood pH, regulating body temperature, hormone levels, etc. And you can see this as an example of the negative feedback here. Now, a couple of things to keep in mind. The feedback can be very short or very long in its loops. For example, the stimulating hormone that's released from the anterior pituitary that would act on the endocrine gland can also have a short loop and feedback to the hypothalamus, thereby blunting out the releasing hormone. Or you could have the actual hormone, which is produced by the endocrine gland, have several long loops. It feeds back and inhibits the anterior pituitary as well as the hypothalamus. Okay? The positive is a little different. Here's what we've got. Of course, you're going to have the hypothalamus and then the anterior pituitary, and then, of course, you're going to have your endocrine gland as your target organ, which will then produce the hormone. The hormone will usually have a positive influence back to the anterior pituitary. So building up increased levels of estradiol will increase the anterior pituitary, which will then affect the endocrine gland, which will cause an increase of even more release of estradiol. Actions are going to basically increase what is called nerve impulses from the cervix transmitted to the brain. What's also going to happen is there's a little part here that is simplified here, but I'll expand upon it. Oxytocin, causing the contractions, will stimulate the release of prostaglandins, which will then go back and work on the hypothalamus, which will cause more oxytocin to be released, which will cause even more contractions. So in other words, what you have is a positive feedback, a cascade that's going to continue until, how does it end? Childbirth. Now, we talk about all of these different hormones. A lot of times they have assessments that are focused on plasma protein levels, plasma hormone levels. I want to bring up several things that you need to be aware of. You can measure hormones or the hormone breakdown products, the metabolites. They can be measured in plasma, serum, urine. There's been a lot of success with saliva also. And as well as you can also measure a certain amount in feces. Okay? Usually the tests that are done now are immunoassays. Go back 40 years, it was radioimmunoassays, but now we've got these nice, clean, non-radioactive means. The only limitation on the immunoassay is the ability of the antibodies to recognize the antigenic sites of the hormone. Now, it sounds like a mouthful, but I want you to understand this. When we talk about an antibody binding, it's binding to a certain section of that hormone. That has to be unique. Also, the hormone cannot be mutated or changed by its antibody levels or something else like that. Furthermore, the antibodies have to be extremely precise. They've got to be extremely selective. For example, there are some hormones like LH and FSH that are very similar to HCG, which is the protein to determine whether someone's pregnant or not. So how did they work out that? Well, there's two chains. The alpha chains for all of the three hormones that I gave you are identical. It's the beta chain, the second chain for HCG, that is unique, and that's where the immunoassays focus in on. The other thing you got to keep in mind is interpreting the data, because that's going to depend on whether the hormone secretion is pulsatile, in other words, circadian rhythms, whether it's sleep, wake. There is a certain amount of cortisol buildup that occurs, increasing levels, just before you wake up. The lowest level, interestingly enough, is about 4 o'clock in the morning. That's why a lot of commando raids are done around that time, when cortisol, which helps us to adapt to stressful situations, are at their lowest. Also, we have to consider nutritional status. And what I mean by that is, okay, iodine. Does the person have enough iodine so that they're making T3, T4? Glucose. Does this account properly for their insulin, their glucagon levels? Water. Are they dehydrated or hyperhydrated? Potassium ions, calcium ions, all of these play a role. Now, let's take consideration for hormonal measurement interpretations. Let's just take measuring hormones that are part of the hypothalamus pituitary axis, HPA. Hypothalamic, pituitary, and the target hormones, for example, TRH, TSH, T3, T4. Hormone levels can be evaluated with appropriate regulatory factors. Okay? You got to keep this in mind. Is it, you're measuring insulin compared to glucose. You're measuring the thyroid hormone to TSH. A lot of times, to look for thyroid function studies, they measure both T3, T4, and TSH, calcium, and parathyroid hormone. Now, we have to keep in mind that urinary excretion of the hormone or the hormone metabolite over 24 hours with normal renal function is necessary for better half-life measurements. Target hormone excess should be reviewed with the appropriate trophic hormone to rule out ectopic hormone production, okay? This is something I don't want to say it's Roberge's rule, but basically it goes like this. If you have a cell that originally was endocrine and it goes cancerous, it's going to become a hormone-secreting tumor. Example, phenochromosatoma, where you have some of the medullary cells in the adrenal that normally produce, upon proper stimulation, epinephrine. Instead, they're going to be screaming full blast, massive amounts of release of epinephrine. And that can create really serious conditions for the patient. Now, you can have other situations that are unique, and sometimes the cells become hidden, or they will show up with unusual amounts in very inappropriate places in the body. Colon cancer. Sometimes you're going to have some lung cancer cells that are going to keep secreting ADH. Key point. Any hormone producing cell, as I said, that becomes tumor cell will secrete hormones in an uncontrolled mode. Okay, so it's going to be basically all brakes are off of it. It's going to be outside of any negative controls, and it may secrete beyond any formerly pulsatile rhythm. In other words, high in the daytime, low at night, high in nighttime, low in the day. It's just not there. Let's mention to you basically about an immunoassay, ELISA. There's several different types. Whenever we talk about the signal, that's going to be something that's going to be like a fluorescent molecule, etc. When we talk about the hormone, it's going to be the target that we're going to bind to. When we talk about the capture antibody, those are usually on the plate. So we take a sample of serum, add it here. And then what we're going to do is the capture antibody is going to hold on to that hormone. Then we wash clean the plate, okay? And then you add a detection antibody, which is also going to be set to bind to this hormone, but it's going to have attached to it a signal. Now, there's a variety of different approaches to immunoassays: direct, indirect, second, third level. I'm not going to get into a ton of that, but I will give you a simple basic point here. How can you make sure you have the positive effect? Well, you can have basically your controls. So if you basically don't add any serum, you've got your capture antibody. You throw in your antibody that's going to have the detection antibody with the signal. You wash that, and if the signal's still there, fine. If the signal's not there, you know that nothing in that little well has the hormone. You got to have your negatives, you got to have your controls, and you also got to have a positive control. That's one where you deliberately added in some of the wells what you're looking for to make sure that the test itself can detect no hormone, confirm that it will detect the hormone, and then you can do the measurements at the various concentrations for the immunoassay. Okay, and you can see this here. A lot of times you get something like HCG, human chorionic gonadotropin. This is basically for detection of, oh, look, honey, I got the line there, I'm pregnant. And what it is that you have your molecule that you're looking for, you have your solid phase antibody, and then you basically have the tracer antibody with the tag. Now, in some cases, what they do is instead of a tag being fluorescent, it might contain a dye molecule. So as the molecule builds up on the little basically filter, you know that you've got the presence of HCG. Okay? Now, a couple of other things to keep in mind. You have stimulation tests that are designed to determine the capacity of the target gland to respond to its control mechanism. You may have heard of them. They're variety. TSH, you could be using the nutrient glucose or calcium. There's a variety of tests that are out there. The suppression test determines whether the negative feedback mechanism that controls the hormone levels is intact. So you might basically use dexamethasone and then measure for ACTH or adrenal cortisol suppression. You might also have receptor measurement. Now, this is usually you're going to have a tissue sample, though. And this can be kind of tricky because of the fact that, it's one thing to sit there and say, well, I can get a piece of skin, and that's tissue, but that's not what I really want. I'm going to need to get some islets of Langerhans. That might be a little bit more tricky surgically. And cost-wise and everything, it's going to be a little funny. Tumor, a designation of a tumor, for example, you can pull out a biopsy of a tumor. What are you doing? You're measuring the number, affinity, and presence of hormone receptors. So applications, you allow the tumor or the tissue responses to the hormone, for example, estrogen receptors in breast tumors. Okay? It's an interesting sidebar note. I'll just say this. Late 90s going into the early 2000s, suddenly older women wanted to try out and did take either an estrogen patch or birth control pills, even though they were postmenopausal, because they thought, hey, this is basically preventing osteoporosis, or my skin looks better, and I can go down a whole list. Some of them sound very, very fashion-wise or very selfish. I just want to look better and all this other stuff. Some of them had some very legitimate points. They wanted to reduce the risk of osteoporosis. FDA eventually banned this because they said the dangers, the risks, are not outweighing or are outweighed by the benefits. And the risk is that if you have breast tumor or reproductive area such as cervix, uterus, that you're in postmenopausal and then you start feeding it estrogen, this will create situations where tumors will begin to grow very, very rapidly. And therefore, it acts as like gasoline on the fire. So, the use for maybe cosmetic purposes or for osteoporosis prevention was banned. And that came out, I believe, in the mid to late 2000s for that application. Now, let me just throw out to you a couple of points here. Here's an example. If you look here on the x-axis, you see basically time. If you look on the y-axis for this one, you have blood glucose levels. And what you have is a comparison between the patients who had a transplanted, islet cells versus the other patients who had normal controls versus those who were your diabetic controls. As the blood glucose level goes up, it stays up for quite some time, as opposed to those who had a normal, healthy islets of Langerhans and produced insulin in a certain way. You see the blood glucose go up a bit after with insulin. You have a period of time. You have the transplanted, which are down here, and you have the diabetic controls. The diabetic controls are the squares. These are the ones with the transplants. These are the ones with the normal. Normal responds to the insulin within a few minutes after a certain period of time with the meal. And you see the levels are elevated, and then they begin to go progressively down. Here you have an individual that is basically the diabetic controls. They're not making any insulin, and it's flat all the way out for 60 minutes past the meal. There are a couple of situations that I mentioned to you before. The dexamethasone suppression test. As you can see, that's what the DST is, or the ACTH hormone. You can see the normal levels. You can see if you had a hyperplasia or an adenoma or carcinoma, how the cortisol levels are going to be much more elevated. After the DST test, you see how all of them sort of limit themselves. Okay. With normals, you would see the cortisol levels drop. Okay. Also with the ACTH, you see the normal level being 6 to 76. With these various types of cancer, what you start seeing you can see this, and notice it says micrograms in 24 hours. So you have to collect the urine for 24 hours. And you have the levels here at being 20 to 90 micrograms. If you have certain situations of hyperplasia, adenoma, and carcinoma, these are going to be elevated. And remember I said earlier that saliva has become an area where you can really use that as a study tool for assays. In this case, we're talking about micrograms per deciliter, and it's assay dependent on the amount. But you do notice in the saliva that there is increases in the cortisol levels with the different types of cells, with the different types of cancer cells. Okay, now, what are we doing here? You're going to see this slide just about every time. What's going to happen is this. I'm going to do basically either case studies or kind of quiz-like questions every week. As soon as the lecture's posted, you're going to have this assignment posted. But you do need to get that assignment done by the end of the week. It has to be uploaded into the Blackboard by Friday at 11:59 p.m. No late work is accepted. Being graduate students, I think that you would understand that. Basically, I'm going to document on completeness, accuracy, and correctness. Now, if in certain situations you are going to answer questions that require that you did a little research, please cite that and cite it in, I am preferring MLA, but if the college demands APA, you can just cite it at the end of your, I want source citations. If you tell me that the moon is made of green cheese and you don't have yes, I'm being facetious now, and you don't have something that says that, A, yes, I'm going to question your sanity, but B, I can also question whether you plagiarized. Okay? We all get information from some source. Now, all the cases, when we do case studies, are going to be reviewed on the Saturday. Now, I'm already set up a Zoom for 9 a.m. on Saturday. Not going to be that long, about an hour. If, let's say, you do a case study and you submit it, I want you to have a copy of it present so you can correct what you got. You don't resubmit it, but this helps you to review and retain the corrected case study sheets as a review tool for your exam. I'm going to put up on Blackboard some compared vital signs data. Also, I encourage you to look into your book index or appendix, and there is a very nice appendix, normal values of metabolic parameters and tests of endocrine functions. Folks, you're going to get used to that. Now, don't worry, I'm not going to sit there and have you on a test. Give me lots and lots of numbers and think you have to memorize those. Because realistically, if you go into the medical community and they send a sample down and the machine does all the work, but it will print out something saying this is above, this is under, here's the normal values, etc. I want you to be able to interpret that, but not memorize dinky little numbers, okay? But I want you to start getting used to looking at what's normal values and what are abnormal values, okay? I'm going to provide in one of the announcements some other additional parameters, blood pH, things like that. So that's going to help you in the determination for your case study diagnosis, okay? We will meet every week on that Saturday. That means on no later than 11:59 p.m. on Friday, you have to submit your work. Do not email it to me. There'll be a site on the Blackboard that you send it right into. I never take attachments. I never take email assignments because there's no place that I can be absolutely sure, A, that I'm going to be able to convey the grades, etc., and B, unfortunately with attachments, and even unbeknownst to students, they may contain viruses or Trojans or something else like that. I don't need my computer crash because of somebody put something and I opened it up and there we go, I'm gone. My computer's all a fritz. So always put your work up in the Blackboard. In every week, there will be a section that will have what is the assignment? Here is the sheet. Fill it out. It's going to be in Word. Make sure it comes back in Word. Okay? Microsoft Word. If you send it to me in some other version and I can't read that source, whatever, I can't read it, it's a 0. I'm sorry. But you got to realize I've got to have certain demands so I can go through this quickly and then be ready to handle your questions. When we have the Zoom meeting will take maybe an hour. We'll go over the questions or go over some of the quiz points or we'll go over whatever is needed that week. But do attend, because this is also a great place where we can throw questions around and say, I didn't understand this. I want you to understand. That's why you're in this course, okay? So have a good week. The next chapter we're going to focus in is in chapter 2 in your book on the hypothalamus.